Hello, my name is Cassie Davis, and I'm a senior at Ottawa Hills High School. The project I've been working on for the past few months is determining intracellular localization of the ion channel CCH1 through genome editing and fluorescence microscopy. First off, I have an interest in cellular biology as it relates to human health and function, as I hopefully will pursue medicine in the future. And an important part of the function of the cell is cytokinesis, which is the final step of cell division, where the cells actually separate after mitosis. And the failure of this process results in cancer, blood disorders, and other neurological defects. So it's important to study the proper influences and causes that impact cytokinesis as it relates to many diseases. And my lab has just recently discovered that calcium is a central secondary messenger in the steps of cytokinesis that are proper cleavage, ferroingression, and cell separation. So the reason I chose this protein to tag is because it's a homologue for another ion channel protein within humans, so I can study this similar protein but with a cheaper and less time consuming model. And also, I want to say that localization is the accumulation of a protein at a given site, and GFP is a commonly used fluorescent protein. My question that I'll be researching is, can I tag GFP, a fluorescent protein, to CCH1, a putative calcium channel in fission yeast? My hypothesis is that vector DNA containing MTM1 promoter and GFP will be transformed into wild-type yeast cells and attach to the CCH1, the gene encoding the CCH1 protein on the N-terminal. As a result, this new genomic sequence will be amplified and the cell will express fluorescence with the protein. So this is the overall methods of my experiment shown in this flowchart. So first, I amplified vector DNA containing the promoter and GFP through PCR. Then once I amplified that DNA, I ran a gel electrophoresis to check if the correct fragment size of that gene was present. And then after I got a positive gel, I performed a transformation with that DNA and wild-type yeast cells, and I used lithium acetate to make the cell membrane permeable and allow the DNA of the GFP to easily pass through into the cell. And hopefully the gene encoding GFP was inserted into the genomic locus of the CCH1. And then after I performed the transformation, I plated them in SPA5S plates, which is nutrient deficient plates, to allow for sporulation. And I let them grow or sporulate in those plates. And then I replicated them into YE5S plates, which is a nutrient dense plate, and as well as G418, which is an antibiotic marker plate. And then after I let them grow in the plates, I took colonies from the G411 plate and performed a genome prep and isolated the genome from the cell colony. And then I performed another PCR of using primers of the CCH1 GFP construct. And then I am performing a gel electrophoresis to see if the gene or the DNA of this new recombinant DNA is present and it has the correct size and length. And then after I get a positive gel, I will use the confocal microscope and see how the fluorescence looks under the microscope and take observations of where it localizes. These are my results, my data. So I sufficiently amplified the vector DNA of MTM1 in GFP, and then right here, and then I performed the transformation with my inoculated wild type yeast cells, and this shows the transformation in the G418 plate, which means that it did take the plasmid into its genome because it has the antibiotic resistance gene within the wild type genome, which it was not previously. And then after I purified the genome of the selected colonies of 6 different colonies I selected at first, I performed a control PCR to ensure that the genome of my colonies was intact. And then, unfortunately, when I went to use the genomic DNA, as well as I screened more colonies from the YU5S just in case, the gel electrophoresis showed that the DNA was not sufficiently amplified, and so I checked multiple different primers, as well as I screened 20 different colonies in various PCRs, which all the gel electrophoresis kind of looked the same, which no banding. And just in case to check if another error was possibly occurred, I amplified just the GFP in the vector DNA to check for that. I'm going to move back and actually discuss what. So I used different primers to search for different locations of the recombinant DNA sequence, and this one checked for just the GFP, and then the other one checked how the GFP connects to the CCH1, and unfortunately both of them were not there. In conclusion, since my plasmid DNA was sufficiently amplified, there was no error there. But I did note that the sporulation after transformation only took 2 days when it actually takes around 4 days, which is interesting to note because it could have been a false positive of growth. And since I ruled out that the primers were incorrect, and if the vector DNA was contained the GFP construct that I was looking for, I ruled those out, as well as the genome of the cell not being intact. So I came to the conclusion that the overexpression of this genomic construct may be lethal to the cell because of the unusually fast growth of the sporulation of the cell. I have evidence to believe that the overexpression of this ion channel could have overwhelmed the cell and caused a major influx in calcium ions. And since calcium is an essential secondary messenger in cleavage for aggression and cell separation during cytokinesis, it could have caused improper cell separation. It caused inefficient, wouldn't allow time for proper mitosis before the cell separates, and which causes an overpopulation of cells that cannot survive on their own. And in the future, I want to do this exact experiment again, but by starting by thawing out the E. coli bacteria and purifying the plasmid myself, because I used lab stock vector DNA just to rule out any other possible errors and ensure that my conclusion is sufficient. And then I also want to try an experiment using a vector DNA that contains a weaker promoter to potentially see if that affects the viability of this of the cell in its DNA sequence. And the implications of this research is that that potentially the overexpression of ion channel gene encoding protein could result in cancers and other diseases caused by insufficient cytokinesis. So unfortunately, I did not get to the microscopy step of this experiment because there was lack of fluorescence expressed by this cell protein. All right, thank you for allowing me to discuss my project, and I hope everything is well. Thank you.